Hello, everybody, and welcome to this episode of Genomics Lite. My name is Sam.
And I am Jack. We both work in the Wellcome Connecting Science team at the Wellcome Sanger Institute. Our roles are to enable everyone in the world to connect with and learn more about genomics. If you haven't joined us before, then Genomics Lite is a series of short sessions that share the science that happens on our campus, which is all about DNA codes. Thank you for joining.
So in our session today, what we're going to be doing is exploring the world of the microbiome. So now some of you watching today might have heard of this term before. We can often see news articles that are around improving our gut microbiome and things like that. The microbiome is a huge area of research. So today we're going to be narrowing it down a little bit and we're going to be focusing in on the nasal microbiome, the microbiome in your nose. We have two amazing guests with us today, and we'll be introducing them shortly, helping us to explore this wide and varied field, and and look at some of the specific bacteria that are in our noses. But before we get started, let's start with the basics. Jack, tell us, what does this word actually, the microbiome, mean?
Oh, well, thanks, Sam. So this word microbiome, it refers to all the microscopic life forms that coexist in a particular location. So just like large life forms that we can see around us with our naked eyes, different microscopic organisms exist in different places. So if we're talking about the human gut microbiome, that refers to all the microscopic life living in that gut. That might be fungi, it might be bacteria or viruses, and they all live there together. A microbiome will be different in different places. So my microbiome in my gut will be different from my microbiome in my lungs, for instance, or as we will be talking about, in my nose. And they'll be different from person to person. So mine will be different to yours, Sam. These groups of microorganisms can change through time as well, and that might be because of different environmental factors. It might be because I'm exercising more, or I'm eating differently, or I'm taking a particular medication. And that change in my microbiome can have a really big impact on my health. So different particular combinations might keep me healthy, and others might make me sick. But how exactly they do this and why they might do this in one person and not in a different person is a really hot area of research. So one reason that this story is so complex is that microbiomes are huge, and the human microbiome in particular, to give you a sense of scale, my body is made up of about 37 trillion human cells, very big number. But my microbiomes, if we added them all together, contain around 3 times that number just of bacterial cells. So that's without counting all the viruses and the fungi. So there are many, many more non-human cells in and on my body right now than human ones. They are microscopic, of course, but it's been said that if I collected all of my microbiome together, then it would take up the volume of around 3 pints, which is more than a litre and a half for those using metric. So it's really, really big. And we won't, of course, be able to cover all of that in today's session. So, as you said, Sam, we'll be talking about the colonies of the microbiome that live within our nose, and in particular, the bacterial colonies.
Okay, well, let's start this by introducing our first guest then to help us unpick this big field. So it gives me great pleasure to welcome Katie, a staff scientist in the Respiratory Virus and Microbiome Initiative in the Parasites and Microbes group here at the Wellcome Sanger Institute. Now, Katie works in and out of the lab using human respiratory samples to investigate how bacteria, viruses, and humans coexist. So thank you for being with us, Katie. As Jack mentioned, the human microbiome is massive. So your work is a little bit more focused around the respiratory areas. So can you paint us a picture about this microbiome for us? What does it look like?
Thank you very much for having me. On the surfaces, inner surfaces of our nose, so right from the entrance right up until we get into almost our sinuses, we have a layer that's commonly called the mucosal membrane. And along that is a nice layer of mucus. But among the mucus and the hairs are lots and lots of bacteria. Many of them exist in linked up to our human cells, so they're binding to things on the surface of our human cells. Some of them are more free in the mucus. And as you get further through, conditions change somewhat, things get saltier, and the bacteria that are able to live in those environments will change as well. So there's lots going on in there. We see some of the outcomes of this, particularly if you're someone who has got a cold at the moment, you'll know that you're getting symptoms in your nose. That's from a virus that's also clinging onto those human cells and then getting inside. But we know that there's lots of stuff up there, and we're setting to exploring that.
And when you say lots is up there, is there a rough number you can give us, like how big are colonies in our nose?
Well, it varies person to person. The amount of space that's available also varies person to person. So we know that from our sampling of healthy people, we have somewhere in the region of maybe 30 species, and then there'll be lots of each of those living in there for us to have been able to detect them.
Okay. And give us a sense as to why maybe why you're interested in the nose, for example. Why is that a good place to potentially look at? Why is it an interesting area of research for you?
Absolutely. So we're particularly interested from a health perspective. So we know that lots of things that we deem opportunistic respiratory pathogens, so pathogens that do make people seriously ill, but also that we find in the noses of healthy people, are there for prolonged periods before they cause any illness, and in some people don't cause any illness at all. So for us, that's a particular interest in Staphylococcus aureus, which is one specific bacteria. If it's resistant to certain antibiotics, we call it MRSA, which people may have heard of. But this is also true for things such as Streptococcus pneumoniae, which can cause pneumonia, and Neisseria meningitidis, which causes meningitis, is one of the causes of meningitis. So knowing a bit more about what the differences are between those bacteria when they live in your nose and when they're causing illness, or what the differences are between the people who go on to develop illness and don't when they have the same bacteria, is potentially really interesting. We think there's also a role for the other bacteria that are there in this, potentially. We know in the gut, for example, that people who have C. difficile in their gut will probably only go on to develop C. difficile as an infection should you take antibiotics and everything else that's not resistant to those antibiotics be dampened down. That's the moment where we would expect a C. difficile infection to occur.
And I just want to take us a step back in just the terms of those, potentially in healthy noses without infection. You saying, 30 species of maybe different types of bacteria and stuff in that nasal passage. Where are we have likely to have picked up those types of bacteria? Where do we pick those up?
So we will have picked them up probably throughout our life through exposure. We'll have got rid of some of them because they will have elicited a large enough immune effect from us that we've got rid of them, or indeed we've had an infection caused by one of them. We've had some antibiotics, it's cleared it up, but the other things will still be there. Exposures take a number of forms. There is a conversation to be had about what people do and do not stick up their noses. But we know that it's somewhat continuous with the skin, particularly near the entry, so it will be rubbing, things like that. When we get deeper, it will be, to some extent, an ecology question of what can survive the deeper you get in, and how tightly bound things are, because the mucus is being wafted by your nasal cells out, though it may not be able to keep up depending on how well you're feeling. But crucially, I suppose, the there's an ecological network going on. So like we see in a beach, say, where there are plants breaking down some things to create nutrients that other plants are dependent on, we know that there will be a network of metabolism going on in the nose, which means that some things have to be there for other things to have the metabolites they require. So the things that they need to feed on to stay alive.
And you mentioned as well earlier a specific bacteria, Staphylococcus aureus. Tell us what that is. Let's imagine that bacteria. What should we have in our minds when we think of that name when you say it?
Sure. So there's actually some clues in the name of Staphylococcus aureus. Aureus is because when we grow it on certain types of media, it comes up nice and yellow colored, so golden aureus. And they're on a plate, lovely glossy colonies, really nice edges. But from a non-bacteriologist perspective, under the microscope, they're nice round colonies in nice long strings. They are most commonly thought about because they cause infections in people. So both more minor skin infections, things like impetigo. If you've ever had like a hangnail become infected, that very well might have been Staphylococcus aureus, through to prosthetic joint infections, bacteremias, so that's bacteria in your blood, or things like endocarditis, so infections of the valves within your heart. When we get to that end of things, these are very serious infections. And even with antibiotics, particularly for things like joint infections, it can be very difficult to treat those. So it's a very interesting bacteria. It lives on the skin, it lives in the nose, it lives in pigs. It survives very well under a huge range of conditions, actually, which is part of what makes it so troublesome when it gets out of your nose and into something where it potentially shouldn't be.
Yeah, that's really interesting. And you work on a study, the Carriage study that is trying to investigate this a bit further. Is that correct? Tell us a bit about this study and what it's hoping to do.
Yeah, absolutely. So the Carriage study has just finished its sampling phase. So within that, about 25,000 people who had previously had host genetics samples taken have agreed to be part of our study and take a nasal swab for us over 3 weeks and answer lots and lots of questions about their lifestyle and how their allergies, their history of infections. And so we're trying to pull that all together now, because when we got those samples, we looked for Staph aureus inside them. So despite being a genomics institute, in this instance, the looking was us trying to grow them in the laboratory. And we're also now taking those samples and looking genomically at what those are constituted. So both looking for Staph aureus within those that we missed in our culture steps, but really more importantly, looking at the big picture of the other bacteria that are in the nose, and so that we can start to unpick some of the relationships there and then link it back to the lifestyle, we hope. And then from there, link it back to the human data that we have about those people as well.
And before I go on to my next question about sort of like your sort of role and what you do as part of that study, just give us a picture of how much kind of before this study did we know around this type of thing? Is this relatively new information that we're starting to get out, or is it has this type of thing been in process before, if that makes sense?
Yes, so the real kicker with this study is the scale of it, and that we're looking at healthy people where we have such a depth of information. There have been quite a number of studies about Staph aureus. It is a topic of interest. People get sick and die with it every year, and the more we know, the better we can prevent that going forward. But typically the numbers have been in the hundreds previously. And when we know that there's such a diversity, it's quite difficult to interpret that with with real confidence to say, no, this is the only way that this could look. Once you've split that, 500 people into 250 men, 250 women, we know there are sex differences between Carriage of Staph aureus. And then we start to break it down. The numbers are too small for us to feel really confident. So one of the things when we were putting together the questionnaire for the study was that we went and looked at what everyone so far had thought might be producing a signal. And we're now trying to go back and say, with our larger number of people, can we replicate that? So really it's an experiment designed to build upon lots of little parts to try and get a real big picture what's going on. And even then, our big picture is of the UK. We haven't been able to expand past that at this point. Hopefully, at some point, we'll be able to replicate almost the same thing somewhere else, because rates do seem to differ between communities as well.
Okay, amazing. And so going back, tell us, as I introduced you, I said works inside and outside the lab. What does that actually look like? Because that sounded quite, elusive. What does it mean when you work inside and outside the lab?
Absolutely. So what it mostly means is I dash between multiple. No, but so inside the lab, when we first set up the study, we're no longer receiving samples. But when we first set up the study, I was part of the team who were handling the processing of those samples. So receiving them all, taking them through a 5-day culture process, recording all the results, sending things for follow-up testing if we needed to, and doing the DNA extraction that we then pass over to operations here who are all set up to do the sequencing for us, which is one of the many benefits of being here. In terms of outside of the lab, there's a lot of data handling that goes along with this, a lot of logistics, and there's also, as I'm beginning to learn, lots of decontamination work and similar to make the data really usable. So there, I fulfill many roles across a breadth of tasks for sure.
Thank you very much, Katie. I want to bring in someone with a different role on this study now too. So I want to introduce our second guest now, Rashene. Rashene is a postdoctoral fellow working alongside Katie in the Respiratory Virus and Microbiome Initiative here at the Sanger. Rashene is a human geneticist and examines how differences in human DNA affect how a person will interact with that nasal microbiome. It's lovely to have you with us, Rashene. Now, I want to ask you what a human geneticist is doing here in this study of bacterial genomics. So how do your skills in human genetics overlap with this nasal microbiome study?
Yeah, thanks for having me. Yeah, it's a question I often get asked as a sort of secret human geneticist within the Parasites and Microbes program. So my role really is looking at the differences in humans that could affect whether or not they are carriers of this bacteria, because we want to take an overall view of the whole sort of ecosystem going on there. And the bacteria are there, but they're living somewhere. They're living in an environment, and that environment is a person. And we know just from looking at people that everyone is different, and that is true on a genetic level as well. So the sort of theory goes that if we are different genetically, and some people can carry this bacteria and some people can't, and the bacteria is maybe broadly, as a human geneticist, I will say broadly the same. Katie would correct me completely. That maybe it's not just things that are going on in the bacteria which allow it to live in some people and not others, but maybe it's things going on with the people as well. So I look at the people.
I'll leave you to fight out the differences in the bacteria after this call. But let's just zoom in a little on what that means. You've drawn beautifully for us that the people are different. give us a sense of how Staphylococcus affects people differently, and perhaps actually begin with how common it is and how present it is in our different populations.
Yeah, so like Katie was saying before, people can either be asymptomatic, which means they are completely themselves, they have no symptoms whatsoever, or you can get these really severe, life-threatening and deadly infections, and then there is a range of things in between that. And that sounds really scary, but actually 1 in 3 people carry Staph aureus in their nose and are asymptomatic. So, I mean, one of us at least on this call, you know? So it's there, it's in the environment, it's in the community, it's there all the time. And there are maybe certain triggers that would cause it to then cause an infection. But for the most part, it's just sort of living out its life inside everybody's nose. But if we understand why it can live in some people's noses but not others, we might get clues as to why it causes infections in some people and not others. And the great thing about the fact that 1 in 3 people carry it is that you can get a really big sample of that. There's a lot fewer people who get really severe infection, which makes it harder to get those big sample sizes where you can actually see an effect. Whereas if we've got in the Carriage study, we have over 20,000 people. That's a third of those people we know are most likely to carry staph. And then Katie and the team in the lab have like proved that that is the case. They've been able to grow it. So because we've got this big sample, we're able to do this really sort of tangible work on these are the differences between these two groups of people.
People watching will perhaps have seen bacteria being grown before. And I loved that description you gave at the start, Katie, of the golden color of it. That really helps me understand. But they maybe don't have such a good idea of how genomics fits in with our understanding of these bacteria and the differences between the bacteria and the human beings part of this study. Could you just unpick for us what DNA is being looked at here and how, I suppose, are you looking at that different DNA?
So I want to cast the net as broadly as possible because I don't want to make any assumptions about what we might or might not find. Which the studies that kind of Katie was talking about before, certainly in the human genetics realm, have focused on, we think this gene is involved, we'll have a look at this gene in people that carry or don't carry the bacteria. Whereas if you just look at one gene, so one unit that controls, like a protein or something in your body, you're going to miss everything that's not that gene. So you're not going to make any sort of new discoveries. You're not going to find what you weren't expecting. So what we have chosen to do is look right across the human genome. So that is your 22 chromosomes and then the sex chromosomes as well. And we want to cast that net as broadly as possible to try and catch anything that is associated with Staph aureus carriage. So the way that we do that is we sequence people's DNA, and DNA is in all of the cells in your body that have a cell nucleus. And these people were blood donors, so they a tiny little bit of blood was taken to then be passed through a load of processes in the lab, which thankfully I am nowhere near. And people who can do those sorts of things sequence that DNA. And what I get at the end of that is output of basically a huge file of letters of different points along the genome that sort of act like flags to say, this bit, I'm here, this is what I look like. And I then scale that up. So for each individual person in the study at the moment, we have about 9 million of those flags across a person's genome. And then we can look and see which of those flags are more common in people who carry Staph aureus. And that is then where we will look further. If that answers the question. I kind of went off about flags.
So we've got a huge amount of DNA data rather than the specificity that has maybe happened in the past. I wonder if you can tell me, where is the study up to, and is there anything you can share yet about what you think you're learning?
So I would love to be able to say, yes, it's all happening. We've got everything. Here are my
We've solved
results.
it.
We've solved it. It's all done. But A, I'd be out of a job, and B, it's not as simple as that. So as much as so I've been working on this project for about a year now, and a lot of that time has been quality controlling the data. So as Katie was saying about the decontamination stuff with the bacterial data, we also have quality control that we have to do on the human side. It's not a case of reading the sequences off a machine and that's fine, we can move forward. There's a lot of making sure that I can match the lab sample to the person and make sure they're the same, that the person's record that says whether they are biologically male or female matches what their genetics says, and making sure that we have the highest quality data possible to then make those inferences. We're in a really lucky position of being able to combine subsampling of 3 healthy blood donor cohorts into Carriage. So those cohorts are the Compare, Interval, and Strides cohorts based at Cambridge University, and they all have their own little individual quirks in their data processing. So I'm in the process of combining those all together. I have done roughly half of our 20,000 people altogether, and I'm still trying to grapple with the other half. And hopefully, in the very near future, I'll be able to put them all together and see and make a proper answer to that question of what have we found. Because at the moment, because we've only got half of the people that we recruited, even if some results were kind of coming out of that, I'd be very unsure about that because it's underpowered. We've not got as many people. The sample size is a lot smaller. So it goes then back to that question of, well, is this actually by chance? What can we take from this and move forward?
And because you've mentioned the patient cohorts there, are you able to tell us a little more about who are the people behind this data?
Yeah.
Are they, you mentioned Cambridge University there, are they from one of the hospitals here in Cambridge, or what can you say about the people in this study?
So the people in this study are incredible. They're so generous with their time and the fact that they had to swab their nose over 3 weeks, 3 times. And that was before, some of them were before COVID. So it was before nasal swabs became a routine procedure. So I think I can never understate the appreciation that I have for the people involved in these cohorts. So these cohorts are healthy blood donor cohorts. So what that means is they've looked at people, we've just included people in England from all over the country who attend either a blood donation centre, which is the interval cohort, or like the vans that go around rural areas to do blood donations, and that's the Compare cohort. And Strides is kind of also people across the country who are blood donors. And that's really good for a couple of reasons. One, blood donors are generally healthy. So you have a sort of generally healthy population to sample from. Two, blood donors are also incredibly generous from the get-go. So you're a bit more likely to get people involved. And three, there's already a wealth of information that these people have allowed the blood donor studies to access in terms of their even just simple things like height, weight, where they had their blood donation taken. All of that information is collated, and then on top of that, they filled out the questionnaire from us about, do you have pets? Have you got an allergy? Have you ever taken antibiotics? All of those things. So yeah, they're from across England because the the sort of NHS in Scotland and Wales is slightly different, so it's a lot easier to do it just across England. And we have good coverage across England, so it's not concentrated in one area. It is a real spread of our population, which is really nice.
Mm. So I misspoke there, didn't I, earlier? I said the patient cohort, but this isn't a patient cohort. This is healthy people donating their nasal microbiomes. I wanted to ask a question about how this study, or how the genomics of this study has changed the way that we can study this kind of bacteria. You mentioned earlier that, previously maybe people have focused just on one gene. That's not been because those scientists were lazy or anything like that. It's because of the technological challenges of studying whole genomes and full DNA codes. And I'm going to ask this for both of you. How has changes in genomic technologies changed the way that you can tackle this kind of problem, studying something within the microbiome? So Rashene, I'll come to you first on that, but Katie, I'll come to you afterwards.
Yeah, this sort of explosion in sequencing, and I guess you guys must hear it from a lot of the people who come here and say like the power of sequencing now, but really like the power and the cost of sequencing to compared to what it was has just opened up this world of good high quality sequences that could be used for multiple purposes, especially because a human's like my DNA is my DNA and will be my DNA in 10 years time, in 20 years time. So I can really examine a lot of different things from almost one sample if my sequence follows me wherever I go. So in terms of like the sequencing power has got really good, but the other thing that's got incredible is the techniques and the statistical methods that we use to look at this, which, I mean, we could sequence all day, every day, but if we didn't have the stats techniques and methods to interpret that, we'd be a bit lost. So, I mean, I'm not claiming to be a genetic statistician, and I never would, but like those people are incredible. They build these programs and methods and really deeply think about how using more data we can incorporate that together to maybe advance both studies. So you could take a study from sort of cardiovascular disease and look at whether those risk factors also impact another condition, and then you sort of boosted both of them. So it's a constant sort of collaborative boosting, I think, now, whereas before it was so labor intensive to sequence that one gene in 20 people, whereas now it's mind blowing, really.
So that's great. Thank you, Rashene, and a great shout out for the statisticians watching. And perhaps if you're studying maths or statistics at the moment, then keep going. It's the field that will help you to solve these genetics problems.
Please do. We need you.
Yeah. But Katie, we've heard from the human side there from Rashene. It feels like the ideas of the microbiome have just been gaining more and more coverage and more notice over the last few years. No doubt that's because of some great advances in what we know and we can tell about it. How has changes in genomics changed those studies?
Absolutely. And even within the lifetime of the Carriage study, there was some pilot work done on a smaller scale before we dug into this section. And previously we were, for our metagenomics, using something called 16S RNA gene sequencing. So as Rose says, basically sequencing a single gene within our bacteria, within our bacterial population, which had slight differences between species. So if we just amplified the one gene, we could see a whole range of species within that sample. What that allowed us to do was, for a lower input and a lower amount of money at the time, to take a look at the species within our sample, which is the first point of call for any microbiome work. But it doesn't allow you to look at any mobile genetic elements, any of the other genes within that population. So for our Staph aureus, it meant we couldn't necessarily say whether that person had a resistant Staph aureus or a non-resistant, what sublineage within the species that belonged to. So it was quite high level. And for some species, it was very high level. We couldn't get beyond a genus into the species. What we're doing now is something called shotgun, which is basically sequencing everything. And a lot of that has to do with costs coming down, but also to do with, as Rose says, the ability to actually properly differentiate all of those samples back out into where they came from and to assemble something useful from it. So we now have a concept of something called a metagenomic assembled genome. So where we've got basically the full genome of a bacterium back out of what was a mixed sample going in. The other half of that is that though Roe loves the human genome, we unfortunately have it in our bacterial metagenomic samples too. And our ability to really pull that out and get to the what we actually are interested in is much better now. So shotgun sequencing becomes plausible in a way that previously we needed to be going after a gene that was just in our bacteria, so that we weren't just amplifying loads and loads of human genes and nothing that we actually wanted.
Shotgun sequencing, I always think is quite a brutal name for this. Perhaps just briefly, could you say why is it called shotgun sequencing to help people understand that?
It's supposed to sort of indicate that it's a broad spectrum, broad brush, scattergun kind of approach. It's not the name I would have chosen, put it that way.
So rather than just zooming in right on one tiny part of these bacteria, we're looking at a much broader thing. We're saying, let's take a look at everything that's here.
Sample has been
great.
Yeah.
And you've just mentioned a topic there that I think is really relevant to ethics within this area, which is we're studying the microbiome, but that microbiome exists within people, and studying the DNA of that, we're going to catch lots of human DNA as well. Rashene, perhaps could you just tell us a little bit about the ethics of how you work in that environment, and how do we decide what we can publish, what we don't, all of those different ethical aspects of working with human and non-human DNA?
Yeah, I think a really interesting thing comparing bacterial and human genetics that I didn't really appreciate before I started this role was bacterial geneticists and researchers can release bacterial genomes once they've sequenced them, so that everybody has access to them, so that they boost the sort of global science resource. Bacteria don't care where their genetics go. Humans really care because it's such personal information. So we don't and shouldn't release people's genetic information, which is part of that, going back to what Katie was saying about that decontamination and quality control process, is removing those human reads, those human sequences from the bacterial information before it gets released, because we want to protect people's privacy as much as possible. They've been generous enough to either donate a swab or their genetics to research, and we should and really do appreciate how responsible we need to be with that. In terms of how I work with that, there are things, there's a lot of training that goes into, there's a lot of training that goes into understanding the responsibility of looking at and working with genetic information, as well as the way that we store and apply to access that data. So we can't just sort of rock up to a research group and say, I'm going to study these human genetics, I want access to this, this, and this. You apply through ethics boards to say, this is what I want to do with this project, this is what I'm trying to understand, this is the minimal amount of data that I need to make sure that I can do that. So you're doing that constantly at every point. You're trying to say, I don't need all of this excess information about people, I just need these bits so that I can look at just this one section that I'm focused on. And the other thing with that is that we, as part of Carriage, have a data manager who is the person who does all of the linking of information. So as researchers, we never see patient names, we never get full dates of birth, we get ages, but I have no idea who these people are. And that, I think, is really important, that there is a complete sort of anonymization wall between people donating their information and me analyzing it. So I could never go back and say, oh, that's Mrs. Jones who lives down the street. I would never be able to do that. And that's really important, that we keep people's privacy.
And you've teed me up beautifully as I get blinded by this brilliant sunshine that we have on campus right now. That we actually did last session of Genomics Lite. We did a session on why trust science and scientists. So I think that's a really interesting point you just raised there. I'm going to take a slight shift because I've looked at the time and it's flown by. This conversation has been amazing. But we kind of mentioned it around real sort of enjoyment of statisticians and continuing that. I wonder, for some of our viewers, to be able to understand a little bit about how you work in the same team but are on different areas, how you kind of both got into your roles and how you got to where you are. So Katie, I'm going to start with you and tell us a little bit how you got there.
Okay. I did an undergrad degree in cell and molecular biology, so I knew I didn't want to be a biochemist. I knew I didn't want to be a straight biologist. So I went for some sort of middling ground, which is where I've then managed to carve out a little hollow for myself. But I first went into the lab on a proper lab on my year in industry, which I did at Public Health England. Porton Down, working on Group B streptococci, which is also another opportunistic pathogen. So I haven't strayed that far from my first introduction to these things. And I really enjoyed it. I learned a lot. I really enjoyed being able to think about big questions with a team of people who were also really interested in them, and brought all sorts of different knowledge together to think about it. We had immunologists, we had epidemiology folks, and the microbiology team as well. And so when I went back to uni, I knew that that's what I wanted to carry on doing. And postgrad, went out and got a job in a research lab in Cambridge, doing DNA extractions, many thousands of DNA extractions. I learned a lot there about a lot of things, and then came onto the Carriage project and ended up sorting the lab side of things during the recruitment of participants, and now learning some bioinformatics too.
Amazing. And Rashene, how about yourself?
So I knew that I wanted to study biology, but again, I wasn't sure what flavor of biology. So I did biology for my A levels, and I actually had to resit a year of my A levels because they didn't go to plan. I also found out I was dyslexic, so it was kind of a big blow to not go to university alongside all of the people I'd been in school with. But I was determined, so like I knew that that's what I wanted to do. So I went back and I did it, and I got into the university that I wanted to. I went to Reading, much to my mother's dismay, as it was a lot further away from home than she had anticipated. But it was there where I really got interested in genetics. Like I had found it interesting before, but we had a compulsory module in first year that just really sort of ignited my sort of passion for genetics. And then I went on to do my sort of dissertation project about Duchenne muscular dystrophy, which is a genetic condition, primarily in boys. And then I did a master's degree in London with a bit more of a specialty lean, looking at genomic medicine and all of the different ways in which genetic conditions, common and rare, affect the population and how that is studied. And then I went on to do a PhD up in Newcastle. So I bounced around a bit, never getting closer to home. So I was up there and I actually worked on rare mitochondrial disease. So moving down here into this role was the first time I'd really looked at bacteria. I sort of knew them in about just like a general term. And I'd lived through the pandemic along with everybody else. So I feel like we all got a bit more knowledge there. But yeah, stepping into here, like I knew that I was really passionate about what is different in people in health and disease and how do we figure that out, and looking at that genetically. And I think looking at that alongside a bacteria like Staphylococcus aureus, where you have this interplay of these two things that are competing, of like your body wants you to be healthy and maybe an infection, staph doesn't want that, and what is going on at that interface, at that that point between them both, I just think is really fascinating. And the fact that we don't know a lot about it because we've not had the techniques to be able to understand it in that way, is like a really perfect sort of model system to work in. So that's kind of how I've ended up here.
We're still trying to convince Ro that bacteria are great, really.
Yeah.
Well, what a message for us to draw the conversation to a close on. I'm very sorry to say that we're at the end of our time. But I love that you highlighted there as well that the number of unknowns, and that we have this new tool or tool that's growing in power to explore these unknowns. I think that's a great thing for people to take away as well. Science is by no means done. We've got lots and lots still to discover. But thank you very, very much, Katie and Rashene, for joining us for Genomics Lite.
Thanks for having us.
You.
And thank you for those that have been watching the session. We'll be taking a short break before our next one. We'll return in August for our next series of Genomics Lite. So make sure after this video, subscribe to our channel, watch some of our old ones, old Genomics Lite series, get your fix of short content as well. You'll see some familiar faces when you watch those. But yes, do stay tuned for more. And yeah, thank you to both Katie and Rashene as well.